All right, hello everybody, welcome back. We are continuing now this week into Module 4.1. So this week we're going to be diving into mechanisms that sort of drive and move ecological processes forward. So previously we were talking about primarily focusing on ecology and environments and the different levels of biology that we tend to study when we think of ecology. And now we're moving into the mechanisms that drive ecology forward. So we're really going to be talking about natural selection, adaptive evolution, and then later non-adaptive evolution. So one of the ways that we're going to start thinking about natural selection is actually thinking about antibiotic resistance. And this is an awesome study from the South African biologist, Dr. Data, and he's been looking at the rise of multi-drug resistant tuberculosis strains and where they come from throughout the world and how they circulate and move throughout the world. So you can see in this image that there are lots of strains that come out of Russia, for example, and then the strains that are prevalent in the United States actually come from Southeast Asia. So this is a way that biologists sort of start to think about capturing how pathogens are moving and particularly how pathogens are changing. And that's sort of going to be the focus of the lectures for this week. So the driving questions for this lecture are really going to be looking at how do bacteria resist the effects of antibiotics? And then through that lens, we're going to be talking about how populations evolve and what is the role of evolution in antibiotic resistance. So when we're talking about antibiotics, antibiotics are simply a way for us to describe a chemical that either kills bacteria or inhibits their growth in some way. It tends to interfere with the function of essential bacterial cell structures. So these are the primary ways in which an antibiotic can interfere with a bacterial cell. Now we're going to be going into the structural aspects of bacteria in later lectures, but it's enough that you know that antibiotics tend to inhibit various essential functions of bacterial cells. In your book, it gives a great example looking at a type of antibiotic class called beta-lactams. So beta-lactams are antibiotics that interfere with a bacterium's ability to synthesize cell walls. So the ability to create and structure these barriers that are so important to the structure of bacteria. And what you can see is that the antibiotic actually inhibits and interferes with the phospholipid bilayer and causes water to flow into the cell and then ultimately lyse or destroy the cell. So we know that bacteria can emerge that survives these types of antibiotics. So even in something that is so dramatic in terms of lysing those cell walls, bacteria can actually not really learn, but can emerge to survive those types of antibiotics. But how exactly do cells acquire the ability to do this? Well, there's a couple different ways, and one of them is that they can acquire random mutations when the DNA replicates during asexual reproduction. Again, we're going to go in more specifically about these processes in later lectures, but it's enough to know that bacteria reproduce asexually through a process called binary fission. So it's basically just when one cell becomes two daughter cells. And during this process, bacteria can acquire these random mutations, and those random mutations may lead to bacteria or antibiotic resistance. So this is what binary fission looks like. So you can see that you start with your cell wall elongates, this DNA is replicated, and it splits into two daughter cells. Now, acquisition of these new genetic traits can result in antibiotic resistance. So there are several mechanisms in which mutations or new structures of DNA can be acquired by bacterial cells. And one of them is called bacterial transduction and bacterial transformation. And that's when bacteria will actually release strands of DNA. And this DNA may include a trait that may be resistant, that may result in antibiotic resistance. And so that's what actually this actual scanning electron microscope of is what you're seeing here is DNA leaving the bacterium to go join into a new cell. Other ways that this can happen is viruses can actually go inside these bacteria, take up DNA, and then deposit it back into a new cell. This can also happen in something called bacterial conjugation, which again we're going to go into later on. But I want you just to know that there are various mechanisms by which DNA, new types of DNA can be introduced to bacterial cells. So this is a great image that sort of puts everything that we're talking about together. So when we're thinking about asexually reproducing bacterial populations, they divide via binary fission. So if this is a normal individual Staphylococcus aureus cell that does not have any antibiotic resistance, it can replicate, and as it replicates, it may create a new mutation. And this new mutation may result in new genetic information. So that's all that this allele is. It's new genetic information. And that genetic information may actually confer some sort of resistance. Again, this is just the mechanisms by which bacteria can change and potentially acquire resistance. Another thing that can happen is this gene transfer, what we talked about previously, in which DNA can transfer from other bacterial populations into this population of bacteria. And as you continue to evolve, you can get these very distinct differing populations of bacteria, one of which could include this idea to be able to confer resistance. And we're going to talk about this and specifically what this population looks like in the next lecture. So see you soon.